Welcome to today's lab, Lab 2 PCR. For today we'll be using a 0.5 to 10 microliter and 10 to 100 microliter pipette. These require the orange tip box for the small ones and the yellow tip box for the larger pipette. First, we will add 100 microliters of master mix to our sample vials. When you do this experiment, you will instead have small containers of master mix to use in your kit. Once we've added the master mix, our next step is to add 3.5 microliters of CO1 primers. Make sure that when you are using the primers, you use only the very small tips and only the small pipette. Once you have your primers, you will add them to your master mix, and then you will mix thoroughly using the pipette, using a in-out pumping gesture. Make sure that your primers are well mixed with your master mix before you continue on. Now get your small PCR strip tubes and label them with all of the samples that you need. We've got 1, 2, positive control, and negative control. To each of your tubes, you will add 20 microliters of mixed master mix with primers. Even though you see Geo using a single tip for all of these, we suggest that you use separate tips so as to avoid contamination. After we use up the master mix, we should dispose of both the tip and the container. This is the CO1 positive control that we will add to our positive control vial. We will add 5 microliters of it. The positive control should always give you bands when you're doing your gel, even if your extractions didn't go particularly well. Make sure that you don't add positive control to any of your other vials. It only belongs in the positive control vial. For the negative control, we're going to add just some plain water. For our other two samples, our fish 1 and fish 2 that we extracted last class, you will add these to the vials labeled 1 and 2. Once you finish adding all of your reagents to your micro vials, then you're going to pick them up and take them over to the thermocycler. Your TA will add yours and all of the vials from the rest of your class into the thermocycler to run for about 2.5 hours at the different temperatures shown. Don't worry, you don't have to memorize these values now. These samples should amplify for use in the gel's next class.